Okay, guys, so here we are on the very last piece of the blood lecture, and we're going to talk about hemostasis. Let's begin by breaking down those words, because usually if you break down the pieces, you can figure out what it means. So hemo refers to blood. Anytime you see hemolysis, that refers to breaking apart blood. So hemo means blood, and stasis means standing or state or stay. And so what hemostasis is just a fancy way to say blood clotting, where you stop blood from flowing. Okay, so that's hemostasis and blood clotting are the same thing. So the next question is, well, why? Why do we need to blood clot? And to look at that, one of the best ways to look at why is to look at what happens when it does not. Okay, so hemophilia, hema again, blood, philia as in loving, blood loving. What it is a genetic disorder where you have any part of the pathway that causes blood clotting to have a disorder, and it makes it so that you either can't clot at all, or in some cases you just have reduced ability to clot. So it really is sort of like a broad category of disorders that results in a spectrum of severities, from can't clot at all to doesn't clot as well. Okay, so and the problem with not clotting is that you're going to have too much blood flow. So even minor, like a small little bruise, it could just bleed and bleed and bleed, and that can end up being like a life-threatening problem with just even a small little bruise. Okay, so which component of the blood is going to be involved in blood clotting? That would be the platelets, also known as thrombocytes, by the way. And so the platelets are going to be formed from, they're actually cell fragments, and they're going to be formed from in the bone marrow along with all of the other blood cells. They form megakaryocytes first, mega meaning big, and so make these big old cells, and then they fragment across, and that creates the platelets. So the platelets are going to be very, very, very tiny, even smaller than red blood cells. And they're just going to circulate around in the body, so that when you get a damage, you're going to create a blood clot. Now there's three kind of phases to hemostasis, to blood clotting. The first is going to be vasoconstriction, which just means the smooth muscle in the wall constricts to make the blood vessel small. And the purpose of that is just to reduce the amount of blood flowing to that damaged region, so you can reduce the amount of blood loss. So after it constricts, the next thing that's going to happen is to create a platelet plug that's going to be using the platelets. They just sort of glob onto each other to sort of block or plug up the hole. And then that's going to be surrounded with a meshwork. I kind of think of like sort of like a fishnet thrown over it, and that's going to be created from a protein called fibrin, and the fibrin is a plasma protein. So when you think about a blood clot, there's really three parts to it. The first is the platelet plug that comes from the platelets, and then the second is the fibrin mesh that goes over it, and then also you have trapped red blood cells in there as well. Right, so you need to create the platelet plug and then reinforce it before you really have a true blood clot. Okay, so let's talk about how that works, starting with a little bit of anatomy. So if we look at the blood vessel wall, there are three tunics, three layers. If you look at the internal layer, the one that's going to be touching the blood, that's called the Tunica Intima. And the Tunica Intima has a layer of simple squamous epithelial cells that create a nice, smooth physical barrier. And then underneath that, you're going to have some connective tissue. So here is the endothelium, which is just the simple squamous epithelium, which creates that smooth snakeskin, low friction barrier. And then underneath it, you have the subendothelium, which contains loose areolar connective tissue that has a bunch of collagen fibers running all through it. That's going to be important. Okay, so when you have a blood vessel that is not damaged, here are your nice, simple squamous epithelial cells, nice, smooth cells, and you create this smooth physical barrier so that nothing can stick to it. They also secrete vasodilators such as nitric oxide and AMP, and that helps make sure that all the red blood cells and the other platelets can't stick to it. But when you get a damage to the blood vessel, what you've done is when you tear off some of those simple squamous cells, that exposes the loose areolar connective tissue beneath it, which means that you now have collagen just out in the open. And so what happens is this is an inactivated platelet. When those inactivated platelets come in contact with the collagen is like a long string of proteins, and as you hopefully remember, proteins are negative. And so when it becomes in contact with that negatively charged collagen, you're going to activate those platelets. And when I say activate, I mean it physically changes in shape, and they become very sticky so that they can stick to each other, and they get really spiky. So what I like to think of it like the stickers, like if you've ever gone hiking, the little seats that cling to your clothes, those are stickers, and it looks very similar to that. You get these little cytoplasmic extensions off of the surface of the cells, and then they just cling to each other like Velcro, right? So they're just clinging to each other, and they can actually activate more platelets. So it's sort of like a game of tag. So once you activate some platelets using the collagen, that's going to then go out and activate more platelets. They recruit more and more and more and more and more. And so this is one of the rare examples of a positive feedback loop where you just get more and bigger and bigger and bigger. And then that's going to recruit enough platelets to physically clog up the damage, the hole in the blood vessel, and that's called the platelet plug. So are we done yet? The answer is no, we are not. Once you create the platelet plug, you then need to reinforce it with a meshwork that's created from the fibrin. So you need to create that meshwork around it. And in order to create the meshwork, you need to activate fibrin. So please remember that fibrin is a plasma protein, but instead of being in an active form, which would be bad because you don't want a bunch of fibrin clogging up the works, what you do though is have a precursor. So an inactive form of fibrin that's called fibrinogen. And this hopefully rings some bells because remember you have like pepsin, remember that enzyme pepsin? What was the precursor to pepsin? Pepsinogen, right? So hopefully you can kind of link those in your mind. So we have the active form of fibrin, that's what's going to create those meshworks that will surround the blood clot. But the precursor, the one that's just sort of flowing along without clinging to each other, that is the fibrinogen, the inactive version. In order to activate fibrinogen and turn it into fibrin, you need an enzyme, and the enzyme is called thrombin. Thrombin related to thrombocyte and the thrombus, which is blood clot. So now what we need to do, and again, I organize this lecture so it kind of levels up on itself. Okay, so at this point, we now know we need to activate fibrinogen to turn it into fibrin, and we do so by using an enzyme. Now we need to talk about, well, how do you regulate the activity of that thrombin? Because you don't want the thrombin going around and activating all the fibrinogen. You need to make sure that the thrombin is only activating the fibrinogen when it's necessary. Okay, so the way you do that is that the thrombin, the enzyme, also has a precursor. The precursor or inactive version of thrombin, the enzyme, is called prothrombin. So prothrombin is going to be converted into thrombin, and that allows the thrombin to then convert fibrinogen into fibrin, and that's what makes them cling to each other to create the meshwork. Okay? Now, the way to activate prothrombin into thrombin, there are two pathways to activate them. They are called the intrinsic pathway and the extrinsic pathway. So what I want you thinking about is, well, where did those names come from? And hopefully that'll help remind you of how they work. Okay, but both of them, intrinsic and extrinsic, both of them are related to converting prothrombin into thrombin so that you can activate the fibrin. Okay, so let's start with the intrinsic pathway first. The intrinsic pathway is the long way. Okay, so what happens is you have anything that is negatively charged. So that means that if you take blood and you put it into a glass test tube or just put it onto a glass slide, glass is negative. So that would activate the intrinsic pathway. Another thing that activates it is, remember that I was saying that the collagen as a protein is negatively charged. Okay, so that collagen that's sitting around by being exposed from the damaged blood vessel, that collagen can then activate the intrinsic pathway. Now, the reason I call it the long way is because after you activate it with anything that's negative, you're going to have a whole series of factors that activate one thing and then another thing and another thing and another thing. It's a cascade. I don't want to go into that whole cascade. I don't think that's worth our time. But I do want you to know one factor, and that's factor 10. See this X right here? That's factor 10. So factor 10, and the reason I want you to know this is because that factor 10 is that's what's then going to eventually activate the prothrombin into thrombin. Okay? So something negative, a whole bunch of stuff happens. Eventually, factor 10 is activated. That then activates prothrombin and turns it into thrombin. Then that thrombin can then go activate fibrin to create the reinforced clot. Okay, so let's look briefly what happens if you have a series of events. Think of it like a chain of events. If you break any piece of that chain, what's going to happen? If you break the chain, any one of those factors, that can result in an inability or a reduced ability to clot. What is that called? That is a broad range of disorders that are collectively called hemophilia. So remember that hemophilia can be caused by any one of those factors having a problem. If any of them are defective, that would result in an inability or reduced ability to clot. And you're going to have a broad spectrum of severity for that disorders. Okay, so let's look at the extrinsic pathway. So extrinsic as an outside of itself, this is the shortcut. Okay, and instead of being activated by anything negative, the extrinsic pathway is going to be activated by a specific substance, which is called tissue factor. And the tissue factor is going to be secreted by the damaged tissues. Okay, so you have the damaged tissues, they secrete tissue factor. And what that does is it activates just two factors. And again, here's the one you need to know. Okay, so you have factor 7 and then factor 10. So it's much, much shorter, a lot fewer, not the long cascade that we saw before. Once you activate factor 10, that then converts prothrombin into thrombin, and there you go. Now you have your blood clot. Okay, so remember that's the one you need to know for both pathways. So let's talk a little bit about where those names come from. So what does the word intrinsic mean? Intrinsic means that it comes from within, right? So if you have an intrinsic quality, it's just who you are deep in your essence, right? From yourself. So an intrinsic pathway, think about how this was discovered, right? If you take blood and you just leave it out, that blood will clot. So if you take blood and put it into a test tube, it will clot. And if you think about this, and if you're just starting to study this, you think, well, hey, if it's clotting, it must be some sort of intrinsic property that causes it to clot. Later, they realize that it's not really intrinsic. What it is a negatively charged glass. But think about how it would be discovered. Whereas the extrinsic pathway, it requires something from the outside, and that specifically would be tissue factor. Okay, so I want to quickly go back to this picture here. You have the release of that the tissue factor. Okay, so you have the tissue factor that goes to tissue factor number 10, that's the X1, that then activates that prothrombin, turns it into thrombin, that then allows the fibrinogen to be converted into fibrin, and that's what reinforces that clot and allows you to form a full blood clot. Sorry. Okay, so the end result is vasoconstriction first, then your platelet plug. Remember, you have the positive feedback loop, and then you're going to reinforce that using both the intrinsic or the intrinsic, often both at once, to create the fibrate meshwork, and then you have your blood clot. Okay, see you later.